Uh, hi everybody. First of all, I want to say that this session will be recorded. So if anyone don't want to be or cannot assist at this webinar, they will found the recording in our YouTube channel and in other our channels. So, hi everybody. We are GFB Honors. We are a foundation that was born in 2013, and we have the aim to promote and facilitate research in sarcoglycanopathies. Now we are happy to present this new series of webinars that will consist in 3-4 webinars focused in the diagnosis and how to get a proper diagnosis. We're going to start today with the Dr. Leonela Luce that going to present about this importance to have an accurate diagnosis in the sarcoglycanopathies and in general in this rare disease. This is very important because to have a proper diagnosis, it will be the initial step to then accede to the clinical trials. So for that, we have invited today Leonela, that she studied biological science at the School of Exact and Natural Science at the University of Buenos Aires, and she carried a PhD and a postdoctoral research in dysferlinopathy laboratory led by Dr. Florencia Gilberto, located at the School of Pharmacy and Biochemistry in the University of Buenos Aires. And currently, she's working at the Research Associate in the NGS group, led by Anna Toff at the John Walton Muscular Dystrophy Research Center. And welcome, Leonela. And now the floor is yours.
First, but thank you for the presentation and you and the organization for the invitation as well. Can you see the screen? Can you?
Yeah, just another thing that I remember that during the webinar, if you have someone have any question, you can write the questions in the chat, and at the end of the presentation, we will ask the questions to Dr. Leonela. So now, yes, you can start. Sorry.
Perfect. Thank you, Carlos. So the presentation I'm going to talk about, as Carlos mentioned before, is regarding the importance of accurate genetic diagnosis in sarcoglycanopathies, but also a bit more in general as well. So let's start from the beginning. Every genetic diagnosis starts from a clinical diagnosis, and that means that the doctor goes through a process of identifying a condition based on the signs and symptoms that the patient presents, but together with the family history of the same condition that the patient is presenting. And this clinical diagnosis relied on the physical examination, where when related to muscle dystrophies or neuromuscular disorders, would be just to check for the presence of muscle weakness, what kind of muscles are being affected, is there proximal or distal muscle weakness, is it on upper, lower limbs, is there any presence of atrophy, pseudo-hypertrophy, what kind of gait pattern the patient presents, how do they walk, the levels of strength of the different muscles, etc. But also the doctor can request for further tests, like, for example, blood test, and this definitely will ring a bell, but they could ask for CK or creatine kinase, which is an enzyme that is normally increased in the serum of patients with muscle dystrophy. So it's a great indicator, but it could also be other enzymes as LDH, aldolase, transaminases, etc. Nowadays, they can also request for imaging tests like an MRI or a TC, just to understand the level of affection of each muscle and the distribution of affection between the muscles and the body. And lastly, they could also request for a muscle biopsy and biopsy-related tests, meaning those tests that require the muscle from the patient to be done. And this could be what are known like immunohistochemistry, where we use an antibody targeting a specific protein from the muscle. And this way we can detect the location and the presence of the proteins within the muscle, if it's correct or not, if they are there or not. But that is in the muscle, but we can also do it in vitro, outside the muscle, outside the cell, and that will be what we call Western blot. And that kind of test will provide us information about the protein per se, the way we're analyzing. Is it has the same size than the normal protein? Is it smaller? Is it present? Is it absent or not? So with all these things, if we would see like a notebook from the doctors after the appointment with the patient, we will see something like this. Well, for example, for this patient, it has an onset at age 5 years, presents proximal muscle weakness, muscle atrophy, frequent falls, unsteady gait. The CK levels were increased. In this case, in this example, would be 5,000 international units for liter, and it has no family history, meaning that there are no other affected individuals in the family. So what the doctor does with all this information is go through his mind and try to connect all these thoughts and try to find a term that would englobe all these terms. And then, for example, it could be something as broad as a limb girdle muscular dystrophy. And I'm saying that it's really broad because the term limb girdle accounts for all those diseases that present principally proximal muscle weakness, and there are over 40 genes that correlate with this. So it's a really broad term, and this would be a presumptive clinical diagnosis. But maybe if we add another of the tests that I already mentioned, like for example, a biopsy showing absence of sarcoglycan, we can reduce or limit this clinical diagnosis or make it a bit more focused. And in this case would be a sarcoglycanopathy, presumptive clinical diagnosis. And why do we say presumptive clinical diagnosis? Because first of all, we don't know what gene is really involved, but also because there is an overlap between the different signs and symptoms present in these muscle diseases, meaning that many of these diseases present the same signs. So we won't have like a real straightforward clinical presumptive diagnosis. So to show you this, I have an example here of limb girdle muscular dystrophy R3, or the limb girdle caused by sarcoglycan alpha. And all the terms that you can see here are terms or signs and symptoms that correlate with this disease. And if I choose 3 and I check all the other genes that can cause the same symptoms, we will see something like this. So if I choose frequent falls, there are 139 genes that the conditions that they cause can present with frequent falls. But if I correlate it with the presence of muscle weakness, from the 264 genes that generate proximal muscle weakness, we can see there is an overlap between these 2 signs and symptoms of 65 genes. And if I add a third one, in this case, I chose the increased CK levels, I go from 306 genes correlated with this sign to a 44 from the overlap. And only one of them is actually the disease that I was investigating. So we will always have a group of genes that are suspected by the clinician rather than just one. And this can only be solved by the genetic diagnosis. The genetic diagnosis will allow us to confirm or rule out the presumptive clinical diagnosis that the doctor provided. And this will be done by different genetic tests that would allow us to identify changes in genes that cause these diseases, or genes that are related to the development of these diseases. And if these are not good enough reasons to do genetic testing, to pursue a genetic testing, we have other ones, like, for example, the genetics test. It is the way to end up with the diagnostic odyssey as soon as possible. It will only be when we know what kind of gene is affected that we can put a stamp on it and determine the diagnosis of the patient with certainty. And that would be the way to end up this odyssey. Also, we are now in the era of gene therapies, meaning that with the therapy, we are being provided with a functional copy of a gene to a patient which gene is affected. So we need to know what kind of gene we are lacking or is affected because it's not functional, so we can provide like a functional copy of it. So, for example, if me as a patient have alterations in sarcoglycan beta, a therapy targeting or providing me with a functional copy of the sarcoglycan alpha is not going to tackle the problem I'm with, so it's not going to be useful. So I need to have that information if I wanted to be eligible for a specific gene therapy. And the same way, nowadays, clinical trials required to have a genetic diagnosis. I need to know what kind of muscle dystrophy do I have before I can be included or not in a clinical trial, especially if these are gene therapies. And lastly, and we're going to come back to it later, these genetics tests that are going to provide with extremely important information to go through genetic testing, which therefore will allow these carriers to go through family planning. But again, we will go back to it a little bit later. But these are like general things for all muscular dystrophy and even more apart from muscular dystrophy to any genetic disease. But let's talk about sarcoglycanopathies. So sarcoglycanopathies, as I said, are a subtype of limb-girdle muscular dystrophies, which, as I said, is a term that englobes diseases, muscle diseases that preferentially affect proximal muscles, proximal limb muscles. And there's no like one specific phenotype or clinical presentation, but rather there is a broad spectrum of disease that goes from severe, which are the patients that have onset during the first decade of life, and they progress really quickly, and therefore they lose the ambulation relatively early in adolescence or early childhood. But it goes to a mild end of the spectrum of phenotype or clinical presentation, where patients, they sort of only present increased CK levels in the serum, or this could also be called also as exercise intolerance. And these patients generally remained ambulant at 50, 60 years of age. But what's the cause behind the sarcoglycanopathy? And as you might know, the cause are genetic molecular alteration, genetic or molecular alterations in 4 different genes, in any of these 4 different genes, which are sarcoglycan alpha, sarcoglycan beta, sarcoglycan gamma, and sarcoglycan delta. And each of these genes generate what they're called different kinds of limb-girdle muscular dystrophies, going from R3 to R6. And these genes encode to 4 different proteins which have the same name, sarcoglycan alpha, beta, gamma, and delta. And if we see this structure here of the muscle, we can see each one of these circles is a muscle fiber. And these proteins are located in what is called the sarcolemma of the membrane of the muscle fiber. And if we do a zoom in of the membrane, we can see all these proteins, and these 4 in particular are the sarcoglycans, alpha, beta, gamma, and delta, which form a complex together, which is called the sarcolicin complex. And what they do, as you can see here, is interact with many other proteins that it will ring a bell for you, like actinin, dystrophin, dystroglycans, sarcospans, etc., which together form like a major complex, which function is to maintain the stability of the membrane of the fiber. So when we do an exercise, when we do something with our muscles, when we go is from a relaxed to contracted position, and is the alternation between the contraction and the relaxed position, between the contraction and the relaxation, where we can have what is like muscle contraction induced damage. So what they do, these proteins do, is to protect the muscle against this damage. But when any of these proteins are missing, then the complete scaffold of proteins is going to be altered, and therefore we're going to go through damage of the fibers. And another way to see this would be what I mentioned before, which is called an immunohistochemistry performed on a muscle biopsy. So this is what you can see in a control sample, where all these circles are the muscle fibers, as we can see here, right? And the green mark are actually marking the proteins, the 4 proteins that we have here, alpha sarcolicin, beta, gamma, and delta. And we can see the borders of each fiber because the proteins are there located. As I mentioned, this is the sarcolemma, is where we should find the proteins, and therefore is what we can see is just like the borders of these fibers. But this is what we see in the control, in a healthy control. What do we see on a patient? In a patient, we see something like this. We see complete absence of the protein, like in here, when we cannot see any of the fibers because gamma sarcolicin is not there. Or we can see a reduction in the amount of fibers that have these proteins. Like, for example, for beta and alpha here, we see that there are not many fibers that have them, or not as intense as in the control, and therefore the shape of the fibers also varies. You can see here the fibers, the size of the fibers are quite uniform, while in here they varied a lot. So when I told you before that there is a spectrum of disease that go from severe to mild for these conditions, this is what it is related to. So depending on the amount of protein that makes it to the membrane or to the sarcolemma and remains functional, meaning to keep to protect the muscle fiber from this damage, it will depend on what kind of clinical picture do we have. So the more protein or the more functional, still functional protein we have in the muscle, the milder the clinical presentation it will be. But if we have absent protein or like a tiny amount of protein, the more severe it will be. And lastly, as I said, these are genetic conditions caused by these 4 genes. So what is the inheritance pattern? So the inheritance pattern is what they call autosomic recessive, meaning that we need to have 2 pathogenic molecular alterations in the same gene to be affected. And we can see it here. As you might know, the human are digenic, meaning that we have one set of chromosomes that are coming from dad and one set of chromosomes that comes from mom. So for each gene, we're going to have 2 copies, one a maternal copy and another paternal copy. And the example that we have here is for sarcolicin alpha, which is located in chromosome 17. And in affected individuals, we can have 2 different scenarios. One would be to have in both of these copies, the maternal and paternal copy, the same mutation, in this case represented by the same star or the same position of the star. And these are individuals that we call homozygous individuals or homozygous variants in the gene. But an alternative situation would be to have two different mutations in the same gene. And these are represented by two different locations of the star in the gene. So one variant that is coming from the maternal side and the other variant that coming from the paternal side. And when you have two different mutations, what you are is a heterozygote or heterozygous variants. But this means that when we have two variants, right, we are affected. What happens if we have only one variant in a gene? In that case, is what it's called as carriers, meaning that because of the presence of a remaining healthy allele or a healthy copy of the gene, they won't present or they won't exhibit any signs and symptoms, but they will still carry one of the variants or one affected copy of the gene. So this means that if we have a pedigree or a family, this is a family tree, we can see in black two affected individuals which are detectable because of the presence of signs and symptoms. All of the other ones in white, which are the ones that do not exhibit any signs and symptoms, meaning healthy, we don't know for sure which ones of them are or not carriers, which one of them carry one variant in the gene and which ones do not carry one at all. And we will again go back to this a bit later. So knowing all this, how do we perform the genetic diagnosis of sarcoglycanopathies? And we need to address two questions. One is, who are we going to study? And who are we going to study? Well, as I said before, whoever has a clinical suspicion or a clinical presumptive diagnosis of a muscle dystrophy or particularly a sarcoglycanopathy. And most especially, anyone who has family history of sarcoglycanopathy, meaning that there are other affected individuals in the family. And the other question that we can ask ourselves is, how are we going to do it? And that depends of what do we know about the case. So in the first case, if we don't know anything about the parents, meaning that there's no family history of the parent, meaning that I am the first affected individual of the family, then what I need to do is to look for variants. Which ones are my variants? Which gene is the cause of my disease? Because everything is unknown so far. And for that, we do something that is called as a genetic diagnostic algorithm, just a set of steps just to go through them to make it till the genetic diagnosis. But it's different the scenario if we have family history, especially other individuals that went through already a genetic test and we know what kind of variants do they have. So if we are in this scenario, we can just go to the analysis of those particular variants that are running through the family. So this is to say that it's really important that when you go to the doctor in the first place, it is extremely important that you share this information with the doctor and that you gather all this information when you go for the first time. Meaning that try to, if you have other affected individuals, try to get the information about genetic tests and the mutations that were identified in those genetic tests. Or in the case that they were performed and nothing, no genetic diagnosis was performed, that is also very important information. So please provide that information to the clinician and make sure that the clinician will share it with the laboratory that will do the genetic diagnosis. So let's talk about a bit of the genetic algorithm. So, because of the characteristics of the gene that are related to muscle dystrophies, and because it's really easy to perform nowadays a sequencing, and with sequencing, it could be something called as Sanger sequencing, panels, exomes, or genomes. Maybe those terms might ring a bell to some of you. That will be the first step of the algorithm to go through. So what we do in this test is just to screen or look for what we call small changes, tiny changes. And what we do is, by different techniques, go through each letter and read through each gene very deeply. And once we have read or learn the sequence or the letters, each letter of each gene that we are analyzing, we compare it with a human reference sequence to know what kind of changes, what is different between the controls or the reference and the patient. And this is a tiny example here. This is a tiny piece of the sequence of sarcoglycan alpha. And this would be the sequence that the reference or control sequence for that. And this is an invented patient sequence. And we can see that between these two sequences, the only difference is between this G or guanine, which is changed from a T or a thymine. And this is what is called a substitution kind of variant, because the amount of letters of nucleotides remains the same, but one had changed for a different one. And if we analyze the consequence of this particular change, we can see that these three together were encoding for an amino acid called glycine. But instead, when this guanine changed to a thymine, instead it's going to code for a tryptophan, which is a different kind of amino acid, which have different characteristics. And therefore, it might alter the functionality of the protein that is generated. An alternative example of a tiny or small change is a deletion. And in this case, these three Gs present in the reference sequence are missing from the patient sequence, and therefore it would remove, delete it. And the consequence between this is that this glycine would be missing. There will be no amino acid there. And therefore, the only difference would be in the lack of one amino acid. And again, later we need to check what kind of consequences can that have in the functionality of the produced protein. If with the sequencing, we cannot find the cause of the disease or two variants, we can go through alternative genetic test. What, for example, could be for the screening of what they are called copy number variants, or could also be called like deletions and duplications. And a technique that can be used for that is called MLPA. It might also ring a bell for you. It could be also an array. And two examples to make it clear. What we see here are how many times a specific area of the genome, of the DNA, is present on each chromosome. And for example, here we have an invented gene that has 4 exons, from 1 to 4, and that's what we would expect in the reference or the controls. But when we test for the patient with this technique, what we identify is that exon 3 is missing. Then again, that's a deletion. Something got removed from the DNA from the patient. But it could also be the other way around, that in this example gene, in our patient, instead of having one copy of exon 1, 2, 3, and 4, for exon 2, we have instead 2 copies, and that would be what is called a duplication. So once we have done all this genetic testing, we are going to get from this test different variants, and we need to try to address or classify these variants, like try to determine which one of them can be a disease causing and which ones are normal changes. And for that, there are some international guidelines that helped us with us. And these are the guidelines that are generated by the American College of Genetics and Genomics. And these are guidelines that are used in every genetic laboratory. And what they say is that we can, with the information of the variant, we can classify each change, each genetic change into 5 categories: benign, likely benign, variant of unknown significance, likely pathogenic, or pathogenic. So what we need to do for that is to gather all the evidence or all available evidence from the variant. Meaning, as it says here, how frequent can we find this variant in the general or healthy population? How frequently we can find it in the among patients from the same disease? If we have a family with many affected individuals, do all of the affected individuals from the family share this variant or not? And for example, as I showed you before, as I need 2 copies or 2 mutations, one in each chromosome, are these 2 candidate variants located within the same chromosome, or are they in those in 2 different ones, right? All that information, I need to weight it up, and if it's tending to be benign, I can classify the variant among benign or likely benign, and then I can discard it as be the cause of a disease. But the other way around, if the evidence supports that the variant has a deleterious effect, then I will classify it as pathogenic or likely pathogenic, and therefore I can say that this is the cause of a disease or might be the cause of a disease. But I didn't put it here. But if I have like contradicting information, some leading to benign, some leading to pathogenic, or I don't have that many information to move the scale one side or the other, I will remain in what is known variant of unknown significance, which is the same as to say that I don't have enough information so far to classify the variants into to the benign side of the scale or the pathogenic side of the scale. And this, the take-off message of this is that because it depends on the amount of information that we have about a specific variant, we need to reclassify the variants. We need to do this data collection of the variants with time and time together, because this classification might change with time. So if you get a report saying that you have a BUS, from time to time request to have a reclassification of the variant or reanalysis of the variant. So when we have done all this, right, we did the genetic testing, whatever it is, or the sequence, the MLPA, and we have classified these variants into these 5 categories, we can face 3 different scenarios. Of course, the one that we want to solve the case would be to have 2 pathogenic variants in 2 different chromosomes. And in that case, I will confirm the clinical presumptive diagnosis of sarcoglycanopathy. But it could also be that I have no pathogenic variants in this gene, in the sarcoglycan genes, and then I will discard or rule out this presumptive diagnosis, meaning that, okay, because of the overlap of symptoms, the cause might be somewhere else, other genes. But I do need to keep doing tests or reanalyze the data that I have generated, the sequencing data. And the third scenario would be just to find one pathogenic variant, meaning that the techniques that I have done only allowed us to detect one of the variants. Maybe the variant that I have in the other copy of the gene, they were not targeted by the techniques that I have done, and I need to go on with further genetic testing, different genetic testing. And unfortunately, most of those tests will require the muscle we're required to perform a muscle biopsy. But I do encourage you to go on through this genetic testing, because it's not going to be until we find both genetic variants so we can confirm the presence, we can confirm the diagnosis of an autosomic recessive disease like sarcoglycanopathy. So, as I told you before, it's important to bear in mind the carriers. As I mentioned again, if we have a family tree, we can only, by the signs and symptoms, identify the affected ones, here the ones that are in black. But any of these white ones, painted in white, which do not present any muscle affection to be clinically identified, we cannot distinguish between the ones that do not carry variants in the affected genes, to those that carry just one. So once we have learned what gene is affected and what kind of variants are affected in this family, we can go through genetic tests to identify or to classify these healthy individuals between carriers or not. Therefore, in this case, we would do a genetic test and algorithm in one of these affected ones, of affected patients. We will identify one variant, but located between those in those chromosomes in an homozygous, or two different variants, and we would test these variants in all of the other individuals to get something like this, where in this case I generated an heterozygote individual where they carry the red variant that comes from the maternal side and the blue variant that comes from the paternal side, and only the ones that carry all the blue, one or the blue or the red ones are the carriers, also the ones that have the black dot inside. And why is it important to do that? Why is it important to discriminate discriminate is not the right word, but to distinguish which one are carriers than not? And it's because, of course, the carriers, because they have this variant, one of their alleles, they can also transmit it, and they can have kids that are also carriers. But this will depend on what kind of partner they are with. So, for example, if this lady starts dating this, well, sounds so fashion, but starts dating this guy which has no molecular alterations in this gene, then together they will have a 50% risk of having carrier children, meaning that to half of her kids, she will provide this healthy allele or a healthy copy of the gene, but in the 50 remaining percent, she might provide the one with the affected copy, and they will also be carriers. But if alternatively she dates this guy that also carries a variant in the same gene, though the risk of having a carrier child are the same, which is 50%, they would also together have a 25% risk of having affected children, which is exactly what happened here. These two carrier individuals presented, not only had, they could only have a healthy non-carrier kid, but it could also have carriers and also affected individuals as well. So in order to establish in which case we are facing or what kind of couple we are through, I would encourage that if I know that I'm a carrier of a mutation in a specific gene for an autosomic recessive condition, as sarcoglycanopathies, I would encourage my partner to be tested at least for variants in the same gene. So if I do a recapitulation of what I just said, for the carrier detection studies, they should be done in any unaffected family member, especially those who want to have kids or they want to go through family planning. This is why, actually, this is the reason why you should do this. And the ways to do an informed family planning, as I said, is to test the partner of the carrier. And this is especially important in places when there is a high frequency of the condition in certain areas. Like, for example, there is an area in Morocco where there is like an increased risk of sarcoleccan, I think was beta. I don't want to lie. I don't want to lie. But if there is a place where there's a lot of affected individuals, then the carrier risk or the chance to meet another carrier, that would also be increased. And then it's really important that they both get tested. And an alternative way to perform this family planning will be to perform or invasive or non-invasive prenatal testing to see if the embryo has both of the variants and therefore will be affected or not. Or an alternative way would be to perform pre-implantatory genetic testing. And in that case, if I know what kind of variants me and my partner are carrying, then I can search for embryos that do not carry both of them, and only those can be implanted. So I hope I provided like an overall overview of what is the genetic diagnosis and counseling that you should receive, right, if you are facing one of these conditions. And now I'm more than happy to accept any questions that you might have. And let me sharing.
Perfect. Thank you, Leonela, for the nice presentation. And okay, so we have received some questions, and I just want to start with this about the diagnosis, because when we have the diagnosis, many of these new diagnoses, as you mentioned, is done with these all with these screenings that they make these sequencings, and they see not just our gene, so not just the sarcoglycan genes or the muscle genes, but they see a lot of a panel with a lot of genes. So we face that we found many diagnoses where there is a specification for sarcoglycanopathies, but there are a specification for other mutations. So I want to ask you what happened with these other mutations, how important they are, and for example, in a diagnosis where they mention two genes that are mutated, how we know which one of these are the disease that we have.
That's really important questions, really good ones. Okay, I will go part by part. Well, the last part, I will start with the last part already. Well, as I said before, every genetic diagnosis will start with a presumptive clinical diagnosis. So it depends on how broad the clinical diagnosis was, what kind of tests you would pursue, and what also it will depend on the accessibility to genetic testing that you have, I guess. Nowadays, it's more convenient, of course, to do like broadened tests, as you mentioned, like panels or exomes, where you analyze for all the genes that are associated with muscle dystrophies or neuromuscular disorders, even more broadly. And despite that, they have the advantage that you are more likely to solve the case, right? Because you will find particularly a variant in those genes, or you expect to find a variant there. The counterpart is that you might find more that you are looking for. And as you said, you might have variants in different genes. And the real answer between those two is that you need to analyze them hand by hand, the clinical phenotype and the results from the genetic test. And the one who actually will evaluate which of those variants are relevant will be the clinician, because he needs to correlate both things. So if I have variants in a gene, they are doubtful because mostly probably you will get variants of unknown significance, right? And the phenotype, the clinical presentation does not match with what the patient has. I think you will rule it out. Those can be discarded. But if you have a BUS, again, a variant of unknown significant, but the phenotype matches, then I would think that the clinician will associate it and say, like, yeah, this is the way to go. And in those cases, I think the clinician, what it needs to do is to ask the lab to, from time to time, keep checking for the information from the variant to go through from an unknown significance to transform it eventually to a pathogenic, or request the lab if there is any alternative test that can be done. So one thing that I didn't mention, but I did mention the carrier testing, is that sometimes when we do the test, we find two variants, but we are not extremely sure, as you mentioned, if this could be the cause or not. So it might be, it is normal that after the test in the improvement, we will request, like with other samples of other family members, like if there are other affected ones or even healthy individuals, or sometimes we more often request for the parents, right? Because that way we can see if there is, in this case, for autosomic recessive diseases, we can see if one variant is coming from the paternal side and the other one is coming from the maternal side. And that way we can decide if there are both chromosome affected or not. So those kind of tests, which are complementary to the one that we did in the proven, could also help us. Yeah, I think that was it.
Okay.
Did I miss anything?
I think, yeah. Now it's clear. Mhm.
Okay.
Another thing that we always found in our diagnosis is
Yes.
this the frame shift mutation and missense mutation.
Yes.
Can you explain a little bit of this? What does it mean?
Yes, perfect. I actually tried to avoid it in the presentation, the frame
Yeah.
shifts, because I thought they might be really difficult. But yes, definitely. So the first example that I did about substitution, for a small substitution, is what we would call a missense change. So a missense change is, we would expect, for example, in the reference sequence, we would expect a particular amino acid in that position. And when we check the patient, we have a different amino acid in that same position. It's like the example, it was from glycine to a tryptophan. So we have the protein will be the same size. It will have the same amount of amino acids that the referent one, but there will be a different in one amino acid or two, right? And sometimes the presence of those changes might alter the characteristics or the functionality of the protein, and therefore it might be that it doesn't make it to the membrane in the case of the sarcoglycans, or they make it to the membrane but they're not as functional. They do not protect the muscle as much as they should, and therefore we have symptoms. Alternatively, the frame shift variants are a bit more complex, and that would be the case of any deletion, right? Or a change in the amount of letters or nucleotides. So I show you there that there was a limited amount of letters there, but and in both cases, well, in the second case, there were three letters or nucleotide that were missing. And the difference will be if the deletion or the part that is missing or it gets added is multiple of three or not. If it's multiple of three, because each amino acid is encoded by groups of three letters, well, we're going to change in the amount of amino acid. That would be what is called an in-frame variant, right? So we will have a shorter version of the protein, again, that might be not functional, etc., but we will still have protein. But if the addition or the deletion of nucleotides or letters is not multiple by three, that would mean that when we read what kind of amino acids are going to be generated, it's going to shift, it's going to change. So we won't have the same amino acid, but a couple less, but we're going to have from the point of the mutation onwards, we will have all different amino acids until it might come to a moment when we find what we call a stop codon, which is a sign that the production of the protein needs to stop abruptly. And if the protein gets extremely shortened, it stops before it should, it might be recognized as something different, something that shouldn't be there, and it might be degraded. And in those cases, in the cases of a frame shoot, what we normally expect is the absence of the protein. So in those cases, you would expect a phenotype that is towards the severe end of the spectrum.
More severe.
Yeah.
Okay.
So that would be the difference. I hope it didn't make it too complicated. Sorry.
Okay. Now another question that is related with this genetic testing. So now we found many times or a lot of publicity advertising that this genetic test that you can do for your family, and you get I don't know, your family have a little bit of part in Argentina, a little bit in France, a
Not the
little bit.
ancestry kind
So
of test. Yeah.
this it's useful for the genetic testing, or it's another thing?
I would suggest that it's not related. I think when you have like a genetic condition, what you need to go is to a specialized center for these kind of tests. Because first of all, the other thing is a commercial thing. They only analyze for these particular things, and they're not going to go through disease-causing variants. It's not what they're. It's not their aim. And they don't have the ethicals that they require to do so. You won't get the genetic counseling that you will require also if you find it. So that's why they don't look for it. But you also need the expertise that is behind a place that does the genetic testing, the expertise of knowing the variants, knowing how to classify them, what kind of evidence do they need to collect. And again, if they find something, you should always get the genetic counseling. They need to help you to understand the genetic basis of the disease that it was diagnosed. Yeah, you need to get assessed for carrier testing, etc.
Okay. And another question that I found very interesting is that, for example, here in Europe, or many of the countries of Europe, we have access to the public system, the healthcare system. But other people in other countries that doesn't have this possibility to get this genetic counseling or this in the public system, which test did you recommend for these people? And the second question is, if some, for example, what a person that live in Turkey could make some kind of test in another country, for example, in the US, if I sending blood, or which kind of test, and if this test can be done elsewhere
Elsewhere,
of our
yeah.
country.
Yeah, it's tough because, of course, it depends on each country, I guess, and the availability that you have. Nowadays, the test to start with would be any kind of sequencing test. And as we mentioned before, it will be a genetic panel for neuromuscular disorders or an exome, I would say. Specifically because small variants are the most frequently found. Of course, you always have the risk that that's not enough to get the diagnosis and you need to go through all the tests. But that would be like you have the most chances to solve the case afterwards. I think that there are countries where even though they don't do it in their own country, the genetic test, they already have the link with labs from abroad. So if you manage to get to a clinician that has that connections, it would be amazing, right? Try to get something like that, or get to know if there is any company trying to encourage that. In Latin America, there were a couple of I'm not sure if nowadays, but a couple of years ago, there were pharmaceuticals like paying and encouraging for genetic testing, at least for some diseases. Like for example, Duchenne, they were like screening and they were paying, so that was for free and it was available, and they were taking care of doing the transfer of the samples to whatever they need to do. Nowadays, in the center now we have, for example, for Latin America, we have what the project that is called LatinSeq. So we are helping or working together with different centers from Latin America to also provide genetic tests. But I will try to find a way to, indeed, if it's not possible in my own country, at least to make it. Because we already mentioned, right, how important it is to have the genetic diagnosis. So it is the way to go if you want to confirm the diagnosis that you have, the clinical one.
Okay. And linked to that, when you do this genetic test for these screenings, sometimes you just need the blood, but you can it's just that you need the blood, or sometimes it's required these muscle biopsies, or how does it work?
So it depends on the case. So, well, the starting point will be, again, if we're going to do a sequencing test or MLPA, something like that, what we need is DNA. If DNA, we can get it from any kind of tissue. That's why we can access via blood, or we can also use saliva. There are also saliva kits that we can use, or these dry blood test, not only like the needle, but also the dry blood that you can get it from the finger. But the problem will be that if after doing those tests, we still haven't found the variants, or we still haven't solved the case, maybe we need to analyze other materials rather than the DNA, like O2 RNA or just the protein. And for those, what we need to analyze is the affected tissue. And in this case, for muscle dystrophy, the affected tissue would be the muscle. And in those cases, then we do need to go through the muscle biopsy. But again, the idea is to start with somewhere, like a place where we can avoid the muscle biopsy as much as possible.
Okay. Okay, so I think that I don't know if we have questions in the chat, but I think no. So, Leonela, thank you very much for your presentation, for your time and your explanation. That I think it have remained very clear. And now, if somebody wants, or in the future, this presentation will be hang in our channels, YouTube and on the website and other things. So again, thank you very much, and thank you to all the public to join us.
Thank you so much, Carlos.
Okay, bye.
Bye.